It sounds like something from a spy movie. A hidden self-destruct sequence, a kill switch buried deep inside the machine, waiting for a signal. But this isn't fiction, it's happening inside you right now. Billions of times a day, your cells are making a life or death decision. And very often, the decision is death. They flick a switch, they quietly and neatly self destruct. This process is called apoptosis, and it's one of the most important things your body does. Without it, you wouldn't be here. You wouldn't have fingers or toes. You'd be riddled with cancer. Your immune system would attack you. Life, it turns out, depends on a finely tuned program for death. So, what is it? Apoptosis is programmed cell death. Think of it as cellular suicide. But it's not a messy, chaotic event. It's a highly organized, deliberate process. A cell that undergoes apoptosis doesn't just explode and make a mess. That would be bad. An explosion, called necrosis, happens when a cell is injured by a burn or a toxin. It spills its guts everywhere, causing inflammation and triggering an immune response. It's like a building collapsing in an earthquake. Debris everywhere. Apoptosis is the opposite. It's like a planned demolition. The building is carefully taken apart piece by piece. The valuable materials are recycled. The site is left clean. The cell shrinks. Its internal skeleton collapses. Its DNA is chopped into neat little pieces. The cell membrane starts to bubble, breaking the cell into small, tidy packages. These packages are then eaten by your body's cleanup crew, a type of white blood cell called a phagocyte. No mess, no inflammation, just a quiet disappearance. Why would a cell need to do this? Why is a kill switch so essential? The reasons are profound and touch every part of our lives, from the moment we are conceived until our last breath. One of the first and most amazing reasons is for development. When you were an embryo, you weren't a perfectly formed little human. You were more like a sculpture being carved from a block of stone. Your hands started out as little webbed paddles, like a duck's foot. The cells in the webbing between your future fingers received a signal. They triggered their kill switch. They underwent apoptosis and disappeared, sculpting your individual fingers. The same thing happened with your toes. It's the process that separates your lips from your gums, that opens your eyelids, and that hollows out the tubes that will become your intestines. Your brain, too, is a product of this sculpting. In early development, your brain overproduces neurons. Then a massive wave of apoptosis culls the excess, getting rid of any neurons that fail to make the right connections. It's a process of refinement. Death carves us into shape. It's a beautiful, if slightly chilling, thought. A tadpole uses apoptosis to get rid of its tail as it becomes a frog. The cells of the tail are no longer needed, so they are programmed to die. Apoptosis is nature's sculptor. But development is just the beginning. Your body is not a static object. It's a dynamic, constantly changing community of trillions of cells, and like any community, it needs maintenance. Cells get old. They get worn out. They accumulate damage. They need to be replaced. Your gut lining, for instance, is a harsh environment. The cells there live for only a few days before they are shed. They are replaced by new cells, and the old ones undergo apoptosis to make way. Your skin is constantly sloughing off dead cells. Your body produces about 1 million new red blood cells every second. To keep the numbers balanced, about 1 million old red blood cells must die every second. Apoptosis is the system of quality control and population management. It keeps your tissues fresh and functional. It's the ultimate decluttering program, constantly tidying up and making room for the new. Without this constant turnover, our bodies would quickly fill up with old, malfunctioning, and useless cells. Perhaps the most critical role for apoptosis is in defense. Your body is under constant threat, both from the outside and from within. Viruses are a perfect example. A virus is a hijacker. It gets inside a cell and turns it into a factory for making more viruses. This is obviously bad for the whole organism. One of the body's best defenses is for the infected cell to recognize it has been compromised and trigger its own self-destruct sequence. It commits suicide to prevent the virus from spreading. It's a sacrifice for the greater good. Your immune system can also help this along. Certain immune cells, like killer T cells, can patrol your body, checking other cells for signs of infection. If they find an infected cell, they don't attack it directly. Instead, they give it a command. They dock with the infected cell and deliver a signal that says, "It's time to die." The infected cell then obediently triggers its apoptosis program. It's a clean, efficient way to eliminate a threat without causing a lot of collateral damage. This brings us to the biggest internal threat we all face: cancer. What is cancer? At its most basic level, cancer is a disease of cells that refuse to die. They have lost the ability to respond to the signals that would normally tell them to undergo apoptosis. A normal cell has a series of checkpoints. If its DNA gets damaged, it will try to repair it. If the damage is too severe to be repaired, the cell is supposed to trigger its kill switch. This is a crucial safety mechanism. It prevents a damaged cell from dividing and passing on its faulty DNA, which is how tumors start. Cancer cells are cells that have broken this rule. They have mutations in the very genes that control the apoptosis program. So when they sustain DNA damage, they don't die. They just keep dividing damage and all. They become immortal, in a sense. They ignore the signals from their neighbors to stop growing. They ignore the internal alarms telling them something is wrong. The kill switch is broken. A huge part of cancer research is focused on figuring out how to turn that kill switch back on in cancer cells. Many chemotherapy and radiation treatments work precisely because they cause so much DNA damage to the rapidly dividing cancer cells that they overwhelm whatever broken safety mechanisms are left and force them into apoptosis. So how does it work? How does a cell actually pull the trigger? It's not one single button, but a complex network of proteins and signals. It's like a Rube Goldberg machine of life and death with multiple triggers and fail-safes. There are two main paths a cell can take to initiate its own destruction: the intrinsic pathway and the extrinsic pathway. The intrinsic pathway is the inside job. It's triggered by stress or damage from within the cell itself. This could be severe DNA damage from radiation or toxins. It could be a lack of oxygen. It could be the withdrawal of essential growth factors, the chemical signals that tell a cell to stay alive and keep growing. When a cell is deprived of these survival signals, it's like cutting its lifeline. It interprets this as a sign that it's no longer needed or is in the wrong place. All these stress signals converge on the cell's power plants, the mitochondria. Mitochondria are more than just power plants. They are the gatekeepers of the intrinsic apoptosis pathway. They hold the key to the cell's destruction. This key is a protein called cytochrome C. Normally, cytochrome C is safely tucked away inside the mitochondria, where it plays a role in energy production. But when the cell is under severe stress, a family of proteins called the BCL2 family gets involved. This family has two factions. There are the pro-survival proteins, like BCL2 itself, which act as guards on the mitochondrial membrane, keeping it intact and preventing cytochrome C from leaking out. Then, there are the pro-death proteins, like BAX and BAC. When the cell gets a death signal, these pro-death proteins are activated. They overpower the guards. They punch holes in the mitochondrial membrane. This allows cytochrome C to spill out into the main body of the cell. The release of cytochrome C is the point of no return. It's the commitment to die. Once in the cytoplasm, cytochrome C binds to another protein, forming a large complex called the apoptosome. This structure is an activation platform. Its job is to turn on the cell's executioners. These executioners are a family of enzymes called caspases. You can think of them as a demolition crew waiting for the signal to go to work. They exist in all our cells, but in an inactive, dormant state. They are like a set of molecular scissors, folded up and safe. The apoptosome finds the first of these caspases, an initiator caspase called caspase 9, and activates it. It unfolds the scissors. This activated caspase 9 then goes on a rampage, finding and activating millions of other caspases, the executioner caspases. It's a chain reaction, an amplifying cascade. One activated initiator can activate hundreds of executioners, which then go on to do the dirty work. These executioner caspases are responsible for the systematic dismantling of the cell. They are ruthlessly efficient. They chop up the proteins that form the cell's internal skeleton, causing the cell to shrink and lose its shape. They switch on an enzyme that goes into the nucleus and chops the DNA into specific, regular-sized fragments. They dismantle the nuclear membrane. They basically take the cell apart from the inside out until all that's left are the neat, membrane-bound packages called apoptotic bodies ready for cleanup. That's the inside job. But what about the extrinsic pathway? This is the outside hit. This is when a cell is ordered to die by another cell, usually a cell of the immune system. Let's go back to our killer T cell. It's patrolling the body and it finds a cell infected with a virus. The T cell has a protein on its surface that acts like a key, a death ligand. The infected cell has a corresponding death receptor on its surface, which is like a lock. The killer T cell literally bumps into the target cell, and its death ligand fits into the death receptor. This binding event sends a signal directly into the target cell. It's a message that says, "Your time is up." The death receptors on the inside of the cell then cluster together and form an assembly platform. This platform recruits and activates another initiator caspase, caspase 8. Notice it's a different initiator than the one used in the intrinsic pathway, but the end result is the same. The activated caspase 8 then sets off the same cascade, activating the same executioner caspases that will dismantle the cell. The cell has no choice but to obey. The kill order came from the outside, from a legitimate authority, and the internal machinery carries out the sentence. These two pathways are not entirely separate. They can talk to each other. For instance, the extrinsic pathway, triggered by an outside signal, can sometimes activate a member of the pro-death BCL2 family, which then engages the intrinsic mitochondrial pathway to amplify the death signal. It's a system with built-in redundancies, ensuring that when a cell needs to die, it dies. The elegance of the system is breathtaking. But it's also a delicate balance. Life exists on a razor's edge between too much cell death and too little. When this balance is disrupted, things can go horribly wrong. Many of our most feared diseases are, at their core, diseases of a faulty apoptosis program. We've already talked about cancer, the classic example of too little apoptosis. Cells that should die don't. They achieve a kind of immortality and grow out of control. Many cancers develop mutations in a gene called p53. The p53 protein is often called the guardian of the genome. Its job is to sense DNA damage. If the damage is minor, p53 halts the cell cycle to give the cell time to make repairs. But if the damage is too great, p53's most important job is to trigger the intrinsic apoptosis pathway. It orders the cell to commit suicide for the good of the organism. When the p53 gene is mutated, as it is in over half of all human cancers, the guardian is gone. The cell loses its primary safety brake. Damaged cells don't die. They continue to divide, accumulating more and more mutations, leading down the path to a full-blown tumor. Autoimmune diseases are another consequence of too little apoptosis. Part of the normal development of your immune system involves creating a vast diversity of T cells and B cells, each capable of recognizing a different target. By chance, some of these cells will be created that recognize your own body's proteins as foreign. These are self-reactive cells. Normally, these dangerous cells are eliminated by apoptosis during their maturation process. It's a form of quality control, but if this process fails, these self-reactive immune cells can escape into the body. There, they can mount an attack against your own healthy tissues. In lupus, the immune system attacks DNA and proteins from the body's own cell nuclei. In rheumatoid arthritis, it attacks the lining of the joints. These are diseases where the body fails to kill the cells it should have killed. But what about the other side of the coin? What happens when there is too much apoptosis? When cells die when they shouldn't, this can be just as devastating. Many neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's are characterized by the progressive loss of specific types of neurons in the brain. The brain has a very limited ability to replace lost neurons. Once they're gone for good. The result is a slow decline in cognitive or motor function. While the exact causes of these diseases are complex, a growing body of evidence suggests that inappropriate apoptosis is a major player. Misfolded proteins like the amyloid plaques in Alzheimer's or the Lewy bodies in Parkinson's can create a stressful environment for neurons. This chronic stress may be enough to slowly push these precious, irreplaceable cells over the edge and into the apoptosis pathway. The neurons aren't being murdered by an external force, they are being driven to commit suicide by their toxic environment. AIDS is another tragic example of excessive apoptosis. The HIV virus infects and kills a specific type of T cell, the helper T cell, which is crucial for coordinating the immune response. But the virus's direct killing of cells doesn't fully explain the catastrophic collapse of the immune system seen in AIDS patients. It turns out that the virus is much more insidious. The presence of the virus seems to make the entire population of T cells, even those that are not infected, much more sensitive to apoptosis signals. They become trigger happy. So, uninfected bystander cells end up killing themselves off, leading to a devastating loss of immune function and leaving the patient vulnerable to other infections. Even in events like a heart attack or a stroke, apoptosis plays a sinister role. During a heart attack, blood flow to a part of the heart muscle is blocked. The cells in the core of the affected area are starved of oxygen and die quickly and messily through necrosis. But in the surrounding area, the cells are not dead, just severely stressed. This stress is often enough to trigger their apoptosis programs. So, over the hours and days following a heart attack, a second wave of cell death occurs as these stressed but potentially salvageable cells commit suicide. This greatly expands the area of damage. Much of the research into treating heart attacks and strokes is now focused on finding ways to block the secondary wave of apoptotic cell death to save as many of those struggling cells as possible. Given how central this kill switch is to both health and disease, the next logical question is, can we control it? Can we learn to flip this switch on or off at will? This is one of the most exciting frontiers in medicine. The answer is yes, we are beginning to. In cancer treatment, the goal is to turn the switch on. Scientists are developing new drugs that specifically target the apoptosis machinery in cancer cells. For example, some cancers survive by overproducing the pro-survival BCL2 proteins, the ones that guard the mitochondria. This makes the cells highly resistant to apoptosis. New drugs have been designed that block these BCL2 proteins. By taking out the guards, these drugs allow the pro-death signals already present in the cancerous cell to finally take over, punch holes in the mitochondria, and trigger apoptosis. It's a way of convincing the cancer cell to finally kill itself. These drugs can be much more targeted and have fewer side effects than traditional chemotherapy. For other diseases, the goal is the opposite. We want to turn the switch off or at least turn it down. In the case of heart attacks, stroke, or neurodegenerative diseases, the aim is to protect valuable cells from dying unnecessarily. Researchers are experimenting with caspase inhibitors, drugs that directly block the executioner enzymes. The idea is that if you can block the demolition crew, you can stop the self-destruct sequence in its tracks, even if the initial signals have already been sent. By blocking caspases in the hours after a stroke, for example, we might be able to save neurons in the surrounding brain tissue that would otherwise have died. This is a much trickier proposition, however. Apoptosis is a fundamental process. Blocking it entirely could have serious unintended consequences, like allowing damaged or potentially cancerous cells to survive. It's a delicate balancing act. The story of the kill switch inside our cells is a story of duality. It is a story of death, but a death that is essential for life. It is the sculptor that shapes our bodies, the janitor that keeps them clean, and the soldier that defends them from threats. It is a process of destruction that enables creation and renewal. The discovery of this hidden program has transformed our understanding of how our bodies work. It has shown us that cells don't just live until they wear out. They are part of a community, and they are programmed to sacrifice themselves for the good of that community. When this program goes wrong, the consequences are severe. Too little death leads to the chaos of cancer and autoimmunity. Too much death leads to the emptiness of neurodegeneration and immune collapse, but by understanding the intricate molecular machinery of this kill switch, the signals, the gatekeepers, the executioner's ease, we are gaining the power to intervene. We are learning how to gently nudge the balance back toward health. We are learning to tell cancer cells that it's time to die and to tell struggling neurons to hold on and live. The kill switch is not just a biological curiosity, it is a fundamental truth about the nature of multicellular life. And it holds the key to treating some of our most difficult and devastating diseases. It's a reminder that sometimes, to live, you first have to know how to die.